with Dr. Ajay Soni from Pursued Life Science, one of our industrial partners. Welcome, Dr. Soni, and well, the round is
Thank you so much.
All right.
So they invited me to share our way of working. We are working for drug discovery projects in our company. So I think it's like really good opportunity to share way we work, and I really open for any kind of discussion, collaboration after this presentation. But like just to start with, so our introduction, we are a company. We are out of Daiichi Sankyo, Japan. So from Daiichi Sankyo, a couple of us started with this concept, and we are working as per the Japanese way of working, where they divide the different the discovery path. So the same way we are working here. So basically, we are working in the early stage of the discovery till clinical. Right? So we identify preclinical compounds, and that's where we stop. And I understand that most of us working here are very on basic research, but end of the day, we all of us want drug out of it. So here we are. So basically, we are a medicinal chemistry company. Although we do have DMPK and biology labs there in Bangalore. Our chemistry labs are here in Manesar. We help with our collaborator in evaluation of projects for design and synthesis of NCs, analysis of data, if there is structure activity relationship or structure property relationship, but I'm talking about DMPK and all. We do synthesize focus compound libraries, and we solve any synthetic challenges, and if required, we do go for scale up of lead compound as well. If I talk about therapeutic area, our primary focus is on oncology, immuno-oncology, inflammatory, and infectious disease here. We work on different targets, including protein degraders, covalent inhibitors, and different kinase and enzyme inhibitors. Although we are open for anything, but these are the primary therapeutic area that we are working here. So we do have our DMPK capabilities. I won't speak much about it, but if you need any support, you can always approach us. We are very affordable when we work for these DMPK studies. This is just a glimpse of our lab here, for chemistry lab and our scale-up lab. So definitely we work very professionally. We are quality oriented. We understand the speed matter and cost also matter. So we work under total quality management services, where we take care of quality, speed, and result oriented, not just output. And this slide where I want to focus on I thought that I should discuss about this slide here, because this is how we could achieve success in several projects. Somehow I'm seeing some, I don't know, some figures should be there, but anyhow. So when we were working with one Indian company before, and our way of working was very, I would say very flat kind of thing. We start project, we start working on one target, end up getting some in vivo compound, and we keep working on one project for several years without any conclusion. So what we have done now, we have divided different stage of projects, and it starts with the RS0 here. Somehow it's not visible, I don't know why, but in this computer it's not visible. So we start with stage 1, 2, 3, 4, and 5 here. So in stage 1, we just first thing evaluate the target. When I say when we evaluate the target, we evaluate everything, evaluate all reference compounds, we test them in-house and see whether we are getting the same result was reported in literature or not. And then out of that one, which way we should go after. And that is the first stage. And if we could not go ahead, I could not get the good results, we just take a decision for no go here. And same way in the stage 2, we do the initial SAR, and we see the tractability of the compounds, like SAR compounds are showing activity or not. And there could be 5 series of compound, out of which we need to choose 2 or 3 series of compounds. We filter out there, and there on we move to the further stage 3. And before we go to stage 3, actually, in fact, we to check the in vivo activity of the compound, that whether we'll achieve our target with those working on those leads or not. So then we go for lead optimization and preclinical evaluation later on. So this way we have decided this one in different stage of project, and every project has its timeline. So I don't know why nothing is visible here.
It's coming on the screen.
No. Anyhow, so I just show you that one. The first stage is like 6 month time. Second stage is 1 year time, and third again 1.5 year time, like that. So we have divided everything in different stages here, because if we complete 6 month time and we could not achieve this one, we are very much strict that we need to take go no go decision. Otherwise, they know a way we should waste time on that project if we could not achieve this target activity in that much time. And we also mention here some kind of figures here. Actually, this is for our academic colleagues, that what kind of investment you need. So like for initial stage of evaluation, we need around 10 to 15 lakh for just I'm talking about synthetic chemistry and DMPK here. So if you want to start some project, you want to have some reference compound profile and all that. So this is initial budget. But and if you go for stage 2 here, like where you want to do tractability and initial SAR, shortlisting of compound, one can go like this much budget. You should always have about 50 to 75 lakh. You should always plan for when you write your grants. And then we go for further lead optimization and so on. So that budget, you should always know that one, like how much budget we need. And this is as per the academic I am putting. For actually for US clients and also it go 10 times higher. So this is really, I'm telling you, this is the minimum budget one should have when we plan for our work, and that's how the discovery work everywhere. So when I'm talking about pursuit here, definitely we are people from MNCs like Daiichi Sankyo, Tokyo and all. But I think most important part is that we are aligned with current trends in drug discovery. Presently, we are working with all clients in US and Europe. And I've just give you insight about 2 different targets. One is covalent inhibitors, other one is PROTAC. So nowadays, if you see, several companies started working on covalent inhibitor and PROTAC. Why? Because there are several targets we were working on, and we have compounds in clinical trial. But the new target we are aiming for, those are non-druggable targets. And especially that, if you talk about even COVID, so the Paxlovid, that drug from Pfizer oral, that is also covalent inhibitor. And that compound was discovered sometime early in 2000, but they could not take it forward because there was a myth that covalent inhibitor cannot be drug. But in fact, there is a drug. Now there are several drugs. If I show you, with that time from 1890s to 2020, there are several drugs in market, especially kinase inhibitors. If you see later stage kinase inhibitors are like they were facing issue with resistance and all that. But once they converted that one into covalent inhibitors, they could come out of the resistance issue, and like the compounds are really very potent, and you need really small quantity of compound, and that is the beauty of covalent inhibitor. And if you see overall trend here and different like publications here, you see with that time interest in covalent inhibitor is building up. And if it's therapeutic area, you name any therapeutic area, everywhere you see people started working for these covalent inhibitors. And at Pursuit, we are very good at for covalent inhibitor. In fact, we have done one project with Dr. Dinesh here for one of the client, and we have synthesized 950 compounds in 2 years time, and 3 compounds are in preclinical. And that's the aggression we work with. We have 1 US patent in last 2 years. If I talk about PROTAC, again, PROTAC is against new strategy. When you have a compound, you see that one is not druggable, and you want to convert that one to druggable target. So you want some drug here out of it. So one way is to convert that one to covalent inhibitor, but other way is that you go for this kind of targeted protein degraders. So here, what we do here is that this compound per se have two side chains here. One is the target where you have a drug which hit the protein of interest, and other way you have some ligase inhibitor here, and in between you have a linker here. So what mechanism of this one is to recruit ligase for the particular target and degrade the protein itself. And this also very potent way of working. Because if you look at several targets, like even kinases and many cancer targets, now people have come up with this kind of protect strategy. And if this concept was introduced in early 2000, and if you see in 20 years, now around 25 molecules are there in clinical trial. And surprisingly, not many people are working on this concept in India. And if you see here, like recently, these two compounds, one is in phase 3, another one is phase 2. These are for oncology. But when I talk about infection also, the recent article in 23 only, if you see, there is an article where we discuss about role of PROTEC in different infection area also. So you name, and again here, you talk about viruses, you talk about bacteria and parasite. Everywhere there is a scope. And if you talk about virus and bacteria, there is already reports. But in parasite, there is yet to see some publication here. But still, because there are all all ligases are there, and you can always plan to design compound using these PROTECs. And this is very powerful strategy because you need very little amount of drug, and your actually, if you have a target compound for particular your proteins and all, you don't need very good efficacy also. If you have micromolar compound also, if you attach this PROTEC linker and all that, you convert to this one, the compound may become up to nanomolar, and it's very safe because you are not hitting any other target here. It's very targeted protein degradation. So this way, if you look at, this is really powerful technique, and I strongly recommend, if you are interested in this one, we can discuss together, we can plan here together, even for parasite also. And it's not difficult to make compound here. We have synthesized more than 3,000 compound in last 3 years are in this area. And everywhere we do see clients are having their preclinical compounds. And in fact, one of the compound is in clinical now, phase 1. So this is something I wanted to share with you. So this is the design. I just said that one. We need a compound which shows some activity for the protein of interest. We need to find some good like E3L binding substrate, and then we design linkers here, and that's how we make compound. In fact, we are working very closely with Dr. Dinesh, and we have like we have some targets for oncology right now, but definitely we are interested for parasite or anything. If you want some work in that one, like even for kinase, if targets are there in malaria or anything is there, we can always think of, converting any compound into the PROTEC. So this is one, I have hidden everything here. Yeah, so I have hidden everything here because confidentiality is important, but this is what we are working with Dr. Asif here. So we could get a good compound. It's a very small work, we have done it, but we could get one potent compound here.
And we are taking it further.
Okay, thank you.
Further.
Good to know that, yeah. So if you have any like kind of support required for chemistry center, so we have both way of working. We can work as per the like your way. We can go for P4 services. If you want, like we can have a collaborative project where we can, start making some compound initially, and then we can collaborative grant writing and fundraising we can go for. This is what we have. All these people are there. Manoj is my partner here. Mark is there in US. He is very good for investment finding. If we have some project, we can get investor from US very comfortably. And Malini is there. She's heading our biology. She has 30-year experience working in Glenmark, and even she was in US for some time. Again, Jadip is there in US, and he's there. Rajiv also there in US. He is taking care of our US counterpart. Thank you so much.
Thank you, Dr. Soni. We have questions. I'll start the ball. Oh, maybe yes.
Now there are a lot of programs where you can in silico predict DMPK, ADME, PK profiles. In your experience, how effective are those predictions as compared to what you do in lab?
Predictions are really helping us out. Nowadays, we do see several AI-based predictions are there, and those are very effective. In fact, one of our collaborators in US, they have come up with in vivo PK predictions also for mice, and they can correlate very well from mice to animal to human data. seems to be good, but some like surprises are always there in that discovery, once we go for it and
In
this,
silico, it is.
but I would say you can always feel the trend, how it's showing that this compound is better than this one. How much better, we don't know. That once you do it, then we know. Yeah, same for talks, right? We do have some, if you go for top predicted software, right? It always show toxic, but you know that one, how much window you want. So this is how it goes.
Question from the audience, maybe the students are curious about how to make drugs after they discovered the key enzyme of the century.
Even in molecule, how we decide how much costs to synthesize it?
So this is predicted cost, but we know that one, like if we start from
Enough steps involved or?
number of steps involved, in general number of steps involved, and the chemical cost also. But what we do in India, basically we start from very cheap raw materials to make it very cost effective. Otherwise, if we start buying everything, there's no point of having Indian benefit.
Mainly we are importing.
Yeah, but we are now.
That's something here, it will be very easy.
They are really reliable. We have been working with them for a long time.
When we have a scaffold, a known scaffold, can you diverse the scaffold into a larger family of things?
Yes, that's what we do.
Okay, now, when you start with like products that are under, say, protection, something like that, you've heard about MMB, for example.
Yeah.
They give away compounds from already existing. Starting from that, and is there any legal thing that blocks you from actually working?
Believe me, we are very good in patent busting. You give me any compound and give me just time, 6 months time, we'll come up with a patentable scaffold out of that.
Sounds very promising.
In fact, I tell you, like one of our experience, one of the compound we are working for leucine transferase, okay? Company had taken very wide patent claims. No one in the world dared to touch that one. And this was the challenge. We made only 35 compounds, we had preclinical compounds in hand. And it's a patentable.
Fair enough.
So if you have anything, we are happy to support you in that.
You know already. All right, if no other question, no? We'll thank our speaker again.
Long live India and France friendship.